키보드 소리 안 들리게.
If you got a phone down here, please come get it and take it. I don't want them down here anymore. Sorry. I'm not going to have these things going off during class. So if you got a phone here, you guys are welcome to record the lectures, keep them on, but there's just too many alarms, too many phones going off. Enough, okay? So if you got a phone over here, just hang on to it. You can record. I understand from everybody you can pick them up fine sitting even up there. I'll yell even louder. Unless it's okay? Anyway, all righty. So, if you want to look and see firsthand the connection between cell signaling and cancer, there you go. And guess what? That's an oversimplified version of what it entails. If you really want to take a look and see all of the components and how integrated all these different signaling pathways are in controlling cancer, basically take a look at the PDF that I uploaded to Colab that shows a figure of all the signaling pathways that are involved that come out of Weinberg's new edition of his cancer book. And that'll really drive it home. And it's not surprising, okay? Because if you take a look up here, all of these signaling pathways, they all entail changes in cellular physiology in response to extracellular events, hormones, growth factors. And so ultimately, all of those different signaling pathways, even the ones that we talked about already with GPCR signaling, all of those can ultimately come down and they are going to influence cell proliferation. Now, they can do it in a positive way or they can do it in a negative way, but the point is, ultimately, the ability of our cells to divide and proliferate are going to be influenced by all of these external signaling pathways and processes, okay? So it's not surprising when you take a look at them and you follow the lines and the straight lines and the dotted lines and everything, no matter which pathway you're looking at, we'll get to this guy in a minute because that's the one we're going to look at, is you notice one thing. First is they all come into the nucleus, ultimately, okay? Because now what we're talking about is that changes in proliferation are long duration, long term. These are not physiological changes that occur on the time frame of seconds to minutes, like we went through with epinephrine, acetylcholine, adenosine, all of that, okay? The reason we have to go all the way to the nucleus is that we are going to reprogram the genes that are being expressed in the cell. So that's the first thing that you notice is we get to the nucleus because now these signaling events are going to change the proteins that are being produced in the cell. That takes time. It takes time to transcribe a gene, it takes time to splice the RNA, export it, synthesize a protein, get the protein routed to its destination. That's all long term, okay? The second thing that you can see, so not only are we reprogramming the proteins that are being expressed, look over here. Ultimately, we're reprogramming the cell cycle, okay? So everything that we went through over the last couple of weeks about cyclin-dependent kinases, maturation promoting factor, going into mitosis, those are also targets of these signaling pathways. But when you're going to reprogram or redirect the cell cycle, that also takes time. It's a long-term response. It's happening in the nucleus, but it takes a long period of time. So the one thing to appreciate going into this problem of looking at cell proliferation in cancer is that the signaling events persist. They're not hit and run, they're not come and go like you saw with hormonal signaling. So we're looking at something that's a much, much longer duration. The pathways all converge in the nucleus. We're changing gene expression, and we're going to be changing cell cycle progression. Okay? Now, the pathway that we're going to focus on, and remember back how many days it took us to go through GPCR signaling. It took quite a few classes to go through it because there's so many players, it branches off, there's so many separate processes that ultimately come tumbling together that it made it more complicated. This pathway is going to be simpler. In fact, if you take a look at it, it's up there in the blue box. The pathway that we're really going to focus on is the primary signaling pathway that's involved in controlling cell proliferation. And this is proliferation that's triggered or elicited by extracellular growth factors. So anytime you see something with a GF after it, it's a growth factor. Generally, they're small peptides, and they always bind to cell surface receptors. But if you take a look at the pathway here, it's a straight shot from the plasma membrane all the way into the nucleus, where we can change gene expression, and then you can see we can also change cell proliferation. So the ultimate target are going to be the cyclin-dependent kinases. Okay? Now, here's the good news. It's not only a straight shot, almost directly linear, But the sweet thing is, guess what? There's no second messages. We don't have to deal with small diffusible molecules anymore other than the growth factor. So we don't have to worry about cyclic nucleotides. We don't have to worry about calcium, DAG, IP3. Forget about it. We don't have to worry about phosphodiesterases. We don't have to worry about caffeine. Those are all, you don't have to worry about them. Instead, you just have to worry about cancer. And here's why. The pathway, as we come down, is going to be comprised of checks and balances, something we hope still persists in our government. So, but here's what we're looking at. It's going to be a sequence of events. We're still going to have a G protein involved, but it's small, it's simple, it's monomeric, and it goes by the name of RAS. We still have G protein involved. But everybody else that's involved in the pathway is going to be a kinase, including the receptor itself. So what you guys are going to see going through this pathway is, it really illustrates kinase cascade. And what it does is, as we move closer to the nucleus, it's going to switch from a kinase cascade to a transcription factor cascade. So we're going to go from regulating activities to regulating gene expression. If you take a look at the RTK signaling pathway and you ask, well, why do we study it in the context of cancer? This is where, as you can see here, circled in red, it's where all the proto-oncogenes disproportionately lie. Proto-oncogenes are normal genes encoding normal proteins that function in normal cell proliferation. So the word that I use there repeatedly is normal. Okay? They are pro-proliferative components. They're transducing the signal from the growth factor down here to try and kick the star into proliferating the cell. And by doing it, we're kicking the cell out of a G0 arrest, where it's just hanging out doing nothing, and we're forcing it to re-enter the cell cycle. Okay? If you perturb, as you're going to see, if we perturb the activity of any of these little guys here in red, any of those proteins, then what it means is they're undergoing alterations, mutations, that allow them to propagate a signal all on their own. They don't need anything that would normally be required upstream of them. And you're going to see examples of how we generate these conversions of proto-oncogenes to oncogenes. And what it means is, the further down the pathway you go, and you acquire an oncogene, all of the preceding steps are irrelevant. We don't need them anymore. Okay? So it means that as you get closer and closer to the nucleus, closer and closer to these transcription factors, they become harder and harder to target. Okay? So that's one of the complexities that you appreciate. You'll see. The other thing that you notice, the checks and balances come because not every component in the pathway is pro-proliferative. And that is, we've got negative regulatory components that are going to be referred to as tumor suppressors. And pretty much for every oncogene, you're going to find a tumor suppressor whose normal role is to keep that oncogene or proto-oncogene in check, to put the brakes on it. And what you're going to appreciate when we look through this pathway this morning is that you're going to see the cell really doesn't want to do this. Our cells have all these safeguards built into this pathway that says, no, do not proliferate, even from the very, very first step. So the cell is constantly having to overcome these barriers that are set up along the way, whose normal function is to prevent the cell from responding, even normally, to an extracellular growth factor. Okay? And you can see that they're lined up as we come all the way down. And then the other thing that you see is that we don't stop there with the signaling components. We also stop it up here at the regulation of cyclin CDKs. So even the cyclin CDKs that are responsible for driving you through the cell cycle are also subject to negative regulation to keep them from working, to stop the cell from proliferating. And you're going to see that among the proteins that are the most important down here is this protein right here, the p53 tumor suppressor. Because our cells are designed to where, unfortunately, p53 is the linchpin. p53 is going to have multiple functions in terms of assessing the integrity of our genome. p53 has been described as the guardian of the genome. And it's simply because p53, of all the tumor suppressors that you're going to encounter, and we'll talk about, p53 does the most. It's the only one that can directly respond physiologically to the presence of damaged DNA. That's where these mutations that convert proto-oncogenes to oncogenes come from. That's how they got there. So p53 is one of the few proteins that can respond to that damage. And it's going to be the only protein that can do the following. It can stop the cell from continuing to proliferate. It'll stop it from going through the cell cycle. It will promote the activation of genes encoding enzymes that can repair the DNA damage. And finally, it will trip the cell into apoptosis and cause it to commit suicide if the damage is not fixed. Lose p53, and none of those things can happen. And as it turns out then, you can almost think of your cancer cells as being little mutagenesis kits that are turned loose. And they are constantly acquiring mutations that if they confer a selective advantage on this cell, then what you're going to see is that that gives a particular cell an adaptive advantage to be able to proliferate when all of its neighbors can't. So the final point going into this is to appreciate the following. There is no such thing as a cancer cell. They are constantly evolving. They are constantly changing. Their genetic signature is changing and evolving from day one. So the challenge there is if you're trying to treat a particular type of cancer, as you're going to see, you can't treat the cell. You really can't treat the disease. You are actually having to deal with a moving target, and your treatment is going to have to evolve based on what genetic signature is responsible for driving that cell's proliferation and allowing it to survive at that moment in time. And as you're going to see, it's a moving target. Okay? So, with that, let's get started on the pathway. We got to start at the beginning, so we're going to start with the receptors. Okay? The receptors that are most involved in cell proliferation are generally grouped into the epidermal growth factor receptor family. And they go by the acronym HER, H-E-R, for human epidermal growth factor receptor. They don't just bind epidermal growth factor, they bind many, many different types of growth factors, but they're the most prevalent. And they are the ones that cause the greatest number of cells to do the greatest amount of proliferation. Okay? We've got 4 of them, HER1 through HER4. They differ, though, and this is what makes it intriguing, okay? For one thing, 3 of them, HER1, HER3, and HER4, have overlapping specificities in binding different extracellular growth factors, okay? So right there we get specificity in terms of which of these receptors a given cell is expressing will determine what growth factors it's going to respond to and proliferate, okay? But one of them is a ringer, and that's this one, HER2. HER2 doesn't bind any ligand, okay? Doesn't have anything that it binds to. But as you're going to see, HER2 can cause problems. And the way that it causes problems is that the resting state of all of these receptors is that they are monomeric. And binding to their ligand causes them to autodimerize. Okay, so they come together, and as you're going to see, that activates an intrinsic tyrosine kinase activity. So that's why they're called RTKs, or receptor tyrosine kinases, and that starts the pro-proliferative pathway. The problem with HER2 is HER2 can partner up with any of the other 3. So we can make heterodimers. And in essence, what it means is is that normally these individual receptors will only dimerize when there's enough of a growth factor out there to bind two monomers and cause them to come together and dimerize. Well, if you put a lot of HER2 on the surface of the cell, which happens in certain types of cancer, particularly metastatic breast cancer, what happens is the HER2 goes up there and it forms heterodimers with HER2, HER3, and HER4. So it sensitizes cells to where now they will proliferate at concentrations of growth factors that normally aren't high enough to cause homodimerization. So that's one problem. Second problem is HER2, if you overexpress it enough, HER2 can actually form homodimers without any ligand. And it can propagate a signal. Okay? So we've got HER2 as a problem. Now, HER2 is a receptor tyrosine kinase that really doesn't function as a receptor then. The other oddball is this little guy up here, HER3. HER3 is an RTK that doesn't function as a kinase. So at the outset, you're sort of seeing these weird chimeric proteins evolve. Most of them bind a ligand, and they could be a tyrosine kinase. HER2 is a tyrosine kinase without binding anybody, but HER3 doesn't have a kinase activity. It's inert. But what can it do? Well, HER3 can still bind to growth factors. And remember, HER2 can't. So the problem is HER3 can form heterodimers with monomeric HER2. Can't bind anything. Those can come together, and that will activate the kinase domain on HER2. Okay? So what you can see by looking at this, you'd like to think that normally we have this well-behaved, regulated process of controlling cell proliferation where HER1 dimerizes, 2 dimerizes, 3 and 4, and now you realize it's not that simple. But understand the one that causes the greatest amount of problems is HER2. Okay? Because it's the one that if you overexpress it, it's a wild card. It can take any of these other receptors that are inert and now cause them to become pro-proliferative. Okay? What we're describing here, everything that you guys are going to see, we're talking about the HER family, but it's also applicable to all these other growth factor receptors. So we're looking at EGF, epidermal growth factor, but it's also applicable to FGF, which is fibroblast growth factor. We got platelet-derived growth factor, PDGF, and then we also have insulin. Insulin is a mitogen. So the insulin receptor also causes cells to proliferate because insulin's responding to high blood glucose levels. So if there's glucose out there, that's a favorable environment for cells to proliferate. They've got a high carbon source for metabolism. So that is why insulin also promotes cell proliferation. But all of them work exactly the same way. So if you notice that we're kind of flipping between PDGF, FGF, and EGF, don't worry about the GFs because all the receptors work the same way. The way that they normally work is monomers bind the growth factors individually. And once they do that, they form dimers. And it's only the dimeric form now that becomes an active tyrosine kinase. And they go to work immediately. And the reason they do it is that they autophosphorylate, the cytosolic domain. So the cytosolic region of RTKs is the kinase domain. It's a tyrosine kinase domain. So what you see is, as soon as the receptors bind the ligand, they phosphorylate tyrosines on themselves. Right there at the very beginning, we've activated the signaling pathway. All right? We don't need anybody else. Now remember, go back to GPCRs. And the purpose of binding a hormone to the beta-adrenergic receptor is we had to activate an inactive G protein, a trimeric G protein complex. And we did that because the receptor, when it's bound to the hormone, is a GEF. And the GEF is what activated the G protein. We've got to solve that same problem here, guys, but we're not going to do it directly. What we're constructing is a jigsaw puzzle at the plasma membrane. So we have to get to RAS, which is in the GDP form. It's a lipid-anchored protein right here, hanging out. It's monomeric, only one subunit, so you don't have to worry about alpha, beta, gamma. It's just RAS. RAS doesn't talk directly to the receptor. So what we have to do, here's the jigsaw puzzle that we're going to build. We're going to use an adapter protein now. We're not making a diffusible molecule like a cyclic nucleotide, so we don't have that luxury. Everybody has to go by protein-protein interaction. So guess what's an important part of this pathway? FRET. It's all proteins hanging out with each other. So what happens is, it's the phosphorylated tyrosines that create a binding site for our adapter. And because it's an adapter that grabs two things, here's a clear example of biologists actually naming something that makes sense. It binds two things, so guess what they called the adapter? Grab two. There you go. Makes sense. It grabs two things. It's got a region called the SH2 domain, and that's just a common motif that binds phosphotyrosine. So the SH2 domain on GRAB2 binds those phosphorylated tyrosines. It's got an SH3 domain that is now going to hook up the GEF. And the GEF is a little protein called SOS. Now here's where the biologists dropped the ball. Because as you guys know from looking at the book, where was SOS discovered? What organism? Of all the things out there in nature that can give rise to bizarre names that mean nothing, which organism comes to mind? Drosophila. It was discovered in flies. So because of that, it got the name son of sisterless. It's real informative, okay, if you're into Drosophila. But unfortunately, it doesn't tell you what it does. So we go from, okay, receptor tyrosine kinase RTK, it's named what it does. GRAB2, it's named what it does. SOS, just don't worry about the name, it's just a GEF. So SOS binds to the SH3 domain. So right at the outset, we've got protein-protein interaction, protein-protein, protein-protein, and then protein-protein. We're linking all of the pieces together at the plasma membrane. Notice there's no amplification here because everything is stoichiometric. 1 to 1 to 1 to 1. Okay? Now, when you guys think back to GPCR signaling, what was the very last part of that regulatory process we talked about? Anybody remember? I know, it's before the election, so, you know, it was before your memories were scarred. But does anybody remember, what was the very last thing we talked about with GPCR signaling? What do our receptors do if the signal doesn't go away? What does the receptor do if you drink too much caffeine? Desensitize it. Guess what? We can cut to the end right now because desensitization in RTK signaling started already. The very first step, the first binding here, guess what? We're starting to desensitize the process. And that is, as soon as a growth factor binds to the receptor, the cell says, I don't want to go any further. So they try not to. This is why the signal has to stay out there for a long time. So here's what happens normally. Let's take a look at HER1. It turns out that the region in the extracellular domain of these receptors keeps them apart. So the reason they don't homodimerize in the absence of the growth factor, they actually have these little sequences that repel each other. So the reason they dimerize is that when they bind to the ligand, that sequence has sort of changed its conformation, and the two come together. So now you've got a dimer that's bound to a ligand. Up to this point, if you were measuring the half-life of the free monomers on the plasma membrane, they have a half-life of about 10 to 15 hours. That's pretty respectable as far as a protein goes. They're out there, they're pretty stable. And what it means is that these receptors, they do get dragged in by endocytosis, and they go to the lysosome, but at a very low rate. Most of them get recycled. So most of these receptors, about every half hour, they get dragged in by endocytosis, they go to the late endosome, and they get recycled back to the plasma membrane, just like the LDL receptor. Look what happens after they bind the growth factor. Things change. The difference here is now they're dimerized. The other difference is these are phosphorylated. So now we've got two competing events that are going on at the same time. One pathway is saying, bind Grab2, bind SOS, let's kick the proliferation pathway over here. The other is saying, I don't want to do that. So what happens as soon as you bind your growth factors, now look at the rates of endocytosis. They go up tenfold. So instead of being endocytosed every half hour, now they're endocytosed every 3 minutes. Instead of most of them being recycled to the plasma membrane, now they go to the lysosome at the very first step. So what happens is that in the presence of a growth factor, the cell is already trying to ignore the signal. It does not want to proliferate. And it immediately starts dragging its receptors off of the plasma membrane, taking them out. Okay? So, and the way it works is, again, the phosphorylated receptors bind AP2 and clathrin, and they come in and make an endocytic vesicle. So how can we tip the balance to get this part of the pathway to work? If we're turning over the receptors much more rapidly than we do without the growth factor, what does it mean? The growth factor has to stay out there. So the first thing that you guys are seeing is that these growth factors don't just come and go like a hormone, like epinephrine. If you want to trigger the cell into dividing, you have to keep the hormone out there for hours. You have to keep it out there constantly, because within a few minutes, the cell is going to have fewer receptors than it did initially. And that is the only way that we can drive the process over towards being proliferation. So there, we've taken care of desensitization. First step, first event, we're already headed to turn the signal off. Okay. But ultimately, if the signal persists, we are going to get proliferation to take place. So here's how we make the jigsaw puzzle work. Okay? So what happens is we're building this complex. So the few of these that don't get endocytosed, during the time that they're there, the receptor binds Grab2, binds SOS, and now we can exchange GDP for GTP. Same old G protein business that we've been talking about all semester. Nothing new here. But see how we've built it? We basically build it by protein-protein interactions. No second messengers, nothing diffusible. Nothing enters the cell. The interaction here only has to be long enough to get the cytosolic protein SOS up to the plasma membrane to where it can engage with RAS. Once we've exchanged GDP for GTP, this process dissociates. And now the SOS Grab2 receptor, if it's still phosphorylated, now this guy can go over and activate another RAS. Okay? So the whole system is contingent on the persistence of protein-protein interactions. So we don't need to care how SOS got its name, but we do need to care how it works. So here's how it functions as a GEF. And this is how all GEFs work. Remember, we've been talking about G proteins. I kept saying, we'll get to the GEF down the road. We'll get to it when we get to RAS. Here's where we got to it. If we take a look at the GDP-bound form of any monomeric G protein, in this case it's RAS, because that's who we're talking about, but it's also true for Rho, RAC, CDC42, RABs, any of them. Every monomeric G protein has this same structure when it binds GDP. And here's the problem. The G protein has a low affinity for GDP, but the GDP is trapped. And in essence, what it is that it's got a little hinge region that's conferred by the switch region right here. Switch 1, here's switch 1, switch 2. Remember, those things have to come together to activate the G protein. Well, switch 1, with the GDP-bound state, it's laying here, and it's blocking the ability of GDP to dissociate. There's a lot more GTP in the cytosol than GDP. It's got a higher affinity for GTP, but the GDP is trapped. So what SOS does, and what every GEF does, by the way, so they all work the same, there's an alpha helical region in the GEFs that come in, and you can see what it does. It places itself in between the nucleotide and the switch 1 region, and it basically pries it open. That's how the GEF works, all of them. So now what happens is the GDP just diffuses away. Low affinity, low concentration. It's immediately bound by a new molecule of GTP. And what that does is that, as you can see, we change the conformation of switch 1 and switch 2. They come together. But the other thing that happens then is that switch 1 snaps back up there again. And now the G protein, the GTP, is now locked. So now we've replaced GDP for GTP. Remember, this is a GTPase. It's got an intrinsic GTPase activity. So that means the duration with which RAS will be active is like any of the other G proteins we talked about. RAS will eventually hydrolyze GTP back to the GDP state, and that changes it from being active to inactive. There is a gap for RAS that accelerates this, and the name of the gap is called NF1, for neurofibromatosis 1. It's a tumor suppressor. NF1 accelerates the rate of GTP hydrolysis by RAS. So it works by shutting RAS off. So now you guys can see already, okay? We propagate the signal from the active receptor to RAS. But RAS is now going to do something, but it will only remain active in sending a signal so long as it has GTP bound. As soon as it hydrolyzes GTP to GDP, it has to go back, re-engage with SOS, and repeat this exchange process again. So that means that for SOS to be able to constantly keep RAS active, we need a receptor that's bound to the ligand. So we have to keep that jigsaw puzzle assembled at the plasma membrane that you guys just saw in order to keep RAS active. Okay? Any questions up to this point? It's pretty straightforward, isn't it? We're just going protein, 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 RAS. Now we go to the kinases, and it's going to get hard. Told you. Okay, now it's not that hard. What makes this hard is the cell is still trying to stop this from happening. Okay? All right, so we've got RAS active. Everybody cool with that? It's over here. We got active RAS. Now we're going to switch to the kinases. There's no second messengers here, so we go straight to the kinase. The kinase that immediately comes after RAS is the kinase RAF, R-A-F. RAF is normally inactive, okay? And RAF is a bimodal protein. It's got two functional domains. It's got a catalytic domain here, and it's got a regulatory domain over this side. The regulatory domain binds to RAS. What we have to do to turn RAF on is that we have to jettison a negative regulator of it. And the negative regulator, sorry to do it to you, it doesn't even have a name. It's got numbers. The negative regulator, it's another tumor suppressor. is this little protein here called 1433. Okay, I don't know if it's some sort of Da Vinci code, but that's what it's called. 1433 is an inhibitor of RAF. It binds to residues that are phosphorylated on the catalytic and the regulatory domain. So to turn RAF on, we basically have to get rid of 1433, the inhibitor. To get rid of 1433, we have to dephosphorylate the serine or threonine on the regulatory domain. That is what active RAS enables us to do. So the inactive form of RAF binds to RAS, and now what that does is that there's a phosphatase in here. They don't know which one, so that's why they don't tell you. And what that does, it dephosphorylates the regulatory domain, and that causes 1433 to dissociate. We've now handed the signal off from the G protein to the first kinase. Notice we're not in the plasma membrane anymore, because RAF can now dissociate from RAS, and it is active. RAF actually remains bound while it's got GTP. When the GTP hydrolyzes to GDP, RAF dissociates. But it's active even while it's bound to RAS. I'd want to point that out, okay? So the minute we dissociate 1433, RAF is now active and we're in the cytosol. And we did that with no second message. Now we've got this kinase cascade. The kinase that gets activated by RAF is this guy here, which is called mitogen-activated extracellular signal-regulated kinase, or MEK, M-E-K. Now, you will also see this referred to as the kinase that phosphorylates the next guy in line, which is MAP kinase. So the other way that and I'm just putting this up here because depending on what you read and what you look at in the literature, you will see this kinase living under another name. And its other name is MAP kinase, or MAPKK. It's just one of these things that people who work on kinases love to do. So, RAF is going to activate MEK. We'll keep it simple. MEK then is going to activate the main player here. The main player in this pathway is the serine threonine kinase, the mitogen-activated protein kinase, MAPK. If PKA lies at the middle of a lot of GPCR signaling pathways, MAP kinase lies in the middle of most proliferative pathways. So mitogen-activated protein kinase gets activated by tons and tons of proliferative pathways. And to get it activated, we go through this process. Now, MAP kinase is normally now going to go and phosphorylate targets to keep the proliferation going. But here's another roadblock the cell throws up. Remember? The cell already threw a roadblock up when you bound the growth factor to the receptor. Said, we're going to shut you down right now. Here's where the cell is also going to try and shut it down. And what happens is MAP kinase phosphorylates RAF. So if you were wondering over here with the inactive form of RAF, where did these phosphates came from? They come from MAP kinase. So MAP kinase is going to actually phosphorylate RAF and recreate the binding site for 1433. So you can see what happens. The cell's gone through all this trouble, we've come all the way down to here, and now we're trying to turn it off at that step. We're trying to shut it down. So now what you can see, in order for RAF to remain active, to keep the process going, who do we need to also have active? Who has to be active to basically enable this RAF, which is constantly being inactive? Who do we need to keep this guy active? RAS. What happens if you've got an so now we'll just look forward. What happens if you have an oncogenic form of RAS that can't hydrolyze GTP? That keeps RAF on. So an oncogenic form of RAS Now we don't need, look at all this stuff now. If we have an oncogenic form of RAS, we don't need anything up here anymore. The signaling starts here. And what that means is RAF is not going to be shut down, because even though when MAP kinase comes on and it rephosphorylates RAF, it goes back up here, it's going to engage with the oncogenic form of RAS, that can't hydrolyze GTP, and the signal will get propagated. Okay? So there's a second example where the cell's trying to shut it down. Okay? So here's where we're going to take a look at how does MEK activate MAP kinase, because it's a dual specificity kinase. MAP kinase is normally inactive. Now here's where you got an eerie parallel with a CDK. Remember that CDKs have a threonine and a tyrosine right next to each other. And they can both be phosphorylated by our old friend WEE1. And that shuts it off. We've got the same situation here, only the phosphorylation is necessary to turn it on. So MEK is a dual specificity kinase. And who it phosphorylates, look again. We got a threonine and a tyrosine that are almost next to each other. There's one amino acid in between them. We've got a threonine and a tyrosine, and they both get phosphorylated in order to activate MAP kinase. So here's a situation where you're positively controlling a kinase by phosphorylating it with threonine and tyrosine. It's the complete opposite of what WEE1 does to a cyclin-dependent kinase. Okay? So we're using tyrosine phosphorylation here. Now you've seen it used twice. It activates the receptor. It activates MAP kinase. So now we're almost at the end, I promise. Is that quick? MAP kinase dimerizes and phosphorylates umpteen million proteins in the cell. We only have to be concerned about a couple. Here's where we go to the nucleus. The active form of MAP kinase goes to the nucleus. That homodimer translocates to the nucleus. And when it comes in, it also activates this other kinase, P90 RISC, but don't worry about it. We don't need to concern ourselves with this kinase. Just follow the MAP. So MAP kinase goes to the nucleus. What both of these kinases do is that they're phosphorylating pre-existing DNA binding proteins that are inactive. They're transcription factors. They're transcription factors that are in the nucleus, but they cannot bind DNA unless they've been phosphorylated. So MAP kinase phosphorylates this one over here, called TCF. P90 RISC phosphorylates this protein over here, called SRF. They form a complex, and they bind to 3 important genes. That's all. Here's where we have a requirement for new proteins to be made for the first time. Everything up to this point, these proteins were all here. These proteins were already in the cell. Now we have to activate transcription. And the 3 genes that we're activating are these 3 genes called FOS, Jun, and MYC. The one that is the most important is MYC, MYC. The reason is the target of these transcription factors. So now what we do is we activate transcription. The messenger RNAs go out in the cytoplasm, they get translated into protein. The proteins come back to the nucleus. They have one main target, cyclin D. So think about it another way, guys. You went through this entire elaborate process that's full of roadblocks and it's full of negative regulatory steps to do one thing, to transcribe the cyclin D gene. Well, that's all you need to get the cell rolling from G0 into G1. Because now the cell is going to start accumulating active cyclin D CDK. And that kinase is now going to start rolling the cell back into the cell cycle. So here's how we're doing it. It's the last step for today. Here's what we've got. We've got genes here that we need to activate. The genes require a transcription factor called E2F. E2F is normally inactive because it's bound to another tumor suppressor, RB. RB stands for retinoblastoma because it's the loss of the RB protein that contributes to that particular type of eye cancer, tumors that develop in the retina. RB keeps E2F from working. So what happens is cyclin D CDK, all the growth factor pathway, comes down to make cyclin D CDK active. And its first target to phosphorylate is RB. So what it is that the cyclin D CDK complex is taking out a tumor suppressor by phosphorylating it. It phosphorylates RB, and that causes it to dissociate from E2F. E2F now starts transcribing who? The next genes in the pathway. Cyclin E, cyclin A, and also itself. So what we're going to do here is we're going to ramp up the production of new cyclins and also more E2F. So now what happens is we're getting into a positive feedback loop because the cyclin E CDK comes up here, it phosphorylates RB. So you can see that what we're doing is we're inactivating RB by phosphorylating it. First cyclin D CDKs did it, now cyclin E and A CDKs are also doing it. We're also increasing the production of E2F. So what happens very rapidly now, we have way more free E2F than we have non-phosphorylated RB to keep it shut off. So now what we're doing is that the combination of ramping up the activity of the cyclin E CDK complexes, now we're able to activate enzymes that are required to start DNA replication. You're now at S phase. So everything that you're looking at for what is going on during G1 of the cell cycle, this is what you're doing. You're ramping up the production of enough cyclin E CDK, cyclin A CDK, to basically drive the cell into DNA replication. And once you start, there's no going back. You're now committed to DNA replication. There, that's the RTK pathway. All right? It all comes down and impinges at the cyclin CDKs. So where we're going to start it on Friday now is we're going to go back through the pathway and we're going to look and see where do oncogenes come from, how are they different, and what can we do about it therapeutically. Okay? That's where we'll start it on Friday.